to show you today a part of my PhD in the Center for Macroecology, Evolution and Climate in the University of Copenhagen. And it's an undeveloped part of my thesis, so don't expect conclusions about this. And it's basically about the footprint of climate change in genetic populations. So we know that climate change has a complex interaction with some traits from the species. So for example, we know that climate change is a good driver for population size of species and for the range size of species. We also know that climate change is a good driver for extinction of species. And there is a very big question nowadays, and it's what is going to happen with the species in the future, right? So then there is this big question, what happened in the past then? So a good thing to do is try to see how climate change affects populations in the past, and now try to forecast in the future what will be the responding species. So there have been some studies about the response of species to climate change. Then, for example, Florez-Rodriguez and al. In 2012, they published this paper in Nature, where, for example, for the bison, they reconstruct the species distribution model in 4 intervals of time, from 42,000 years ago to 6,000 years ago. And then we can see that the orange part of the maps is going to a reduction. And then they estimate the area of this simulated distribution of species. And then they found that from the past to the present, there was a reduction in the area of the species. So when they estimated the effective population size based on genetic markers, they found that the pattern in the population size is mirrored by the pattern in the area. So basically there is a good relationship between the area of the distribution of species and the population size of species. And recently, Raquel Garcia and collaborators published this paper in Science, where they said, like, okay, we know that climate change has an effect on species, but there is many metrics that we can estimate for climate change. And one of these metrics is the climate change velocity. Climate change velocity is basically, if we have a grid where we have values for climate, then we can estimate how much climate in a cell of this grid is moving, right? And that will be important because if we assume that the species track the suitable environments, then if you have conditions that are moving very fast, we expect that species have to move very fast to stay in the suitable conditions that they need to have a very good carrying capacity. So, okay, now we know that climate change has an effect in the range size, population size. We know that the range size and population size, they mirror the patterns of each other. So what would be a good scenario to test hypothesis about climate change and species responses? Then it would be the late quaternary. Oops. During the late quaternary, there was a lot of change in climate. So many of the papers published about climate change are related to last glacial maximum, younger dryas, vulnerable erode. So it's a period of time where a lot of climate change happened. And it's a period of time where a lot of species went extinct. Here's some examples of species that went extinct. But then if we have the temporal frame where to go to evaluate the effect of climate change in species, then what will be the kind of data that we need for that? Well, the heterochronous data will be a good data. Let me explain what is heterochronous data. If we have a profile, right, stratigraphic profile, then we can have fossils that are these big dots in the profile. So for these fossils, we can actually extract DNA. So it will be these blue points, the pink with blue here. And we can also have modern data. So basically we have DNA data that it has been sampled many times in the temporal scale. So we'll have a time series of ancient DNA. And then we can have some restrictions about the fossil. For example, if the fossil is stratigraphic dated instead of C14 dated. And then we can have data that is based on ancient DNA and modern DNA. That will give us a time series to explore the genetic parameters of the species through time. So what I did, I compiled a data set for 13 species of mammals. All of them are distributed in the Holárctic region. We can see that, for example, some of them are extinct. There is the woolly rhino and the mammoth. Some of them are big herbivorous mammals. Some of them are rodents. Some of them are carnivora. So we have a lot of variation in the traits for these species. For all of them, we have fossil DNA, and all of them are distributed in the Holárctic region. So what I did was to estimate the population size of species through time. And this is the response of these 15 species. If we see it's a mess, we cannot see any pattern there. We cannot see that, for example, in the last glacial maximum row, they had a drop in population size, things like that. So still, if we see this graph, we will say like, okay, the response is individualistic, right? But what happens when we divide this response or pattern by guilds? Then we can see, for example, that herbivores, they tend to have a drop in the population size to the present. This is past, this is present, this is population size. So we can see that most of the species, they have a drop in population size. But what happens when we see small rodents, then they actually tend to have an increase in the population size. And what happens when we see the carnivora, they actually have a flat pattern in the population size through time. So it's not actually individualistic, the response. It's a response that it should be very clustered by guilds or traits of the species. So let's bring some of the climate here. So I will show you an example of 3 of the species that I have, where I'm going to include a velocity of climate to estimate the population size of species. So this is the actual sampling for the species. So the dots are the points where I have ancient DNA. The colors represent the age of the species, and this histogram is basically the distribution of the data from the 0 to past for the species. So what I did with this point was to extract the velocity of climate. It's not very clear, but I will try to show it. So if we see these bar plots that are here, it's the velocity of climate for every being extracted from the fossil record that I have. So basically, this line here is representing the change in climate for every one of the species. Right? What we see in green is the slope of the population size through time. And what is very nice is that if you compare these 2 lines, they actually see like they mirror each other. So they vary in the same way. So at the first sight, you will say, like, for example, this peak here that is the biggest climate velocity. For the bison, it will be the biggest slope in the population size, right? And it's the same for other species. But what is very weird is that when I do the cross correlation for these 3 species, it turns to be, this is a cross correlation graph that is, so that you cannot see very clearly, but I will try to explain. So basically, what we have in the x-axis is the lag for the 2 time series. If we have the biggest line in 0, it means that you don't have to move any of the lines to have the bigger correlation. But for 2 of the 3 species, we have a -1. What it means that the response species, it happens before the change in climate. So it's actually not a response. And for one of them, the response is in 0. So basically, even though they vary in the same way, I cannot say that it's a response to climate. It's happening before the change in climate. Right. So to summarize, basically, the responses of the species to climate change in effective population size rather than be individualistic response could be clustered by traits, and I have to deal with this -1 lag because it's not a response to climate change. Thank you. Maybe that was too fast. Yeah, you have time for questions.
So of course the other thing going on is human occupation. There's a long debate between overkill and climate. Some of your taxonomy, like we had a rodent or something in there, I'm not sure what it was, would not be subject so much to the human operations of the direct. So do they show a better match to climate change than the taxonomy might be?
Well, there is a, you're completely right, and I don't have any data for humans, so I haven't actually evaluated how humans are affecting the model that I'm using. And, but I know that, for example, humans were exploring more big mammals than small mammals, but still some mammals have been found in this excavation for humans. So I don't really know how much have been the effect of humans in either big mammals or compared to small rodents.
I guess my question is, do small rodents show a better fit to the climate velocity than large mammals?
I haven't evaluated the small ones. That was our last thing to put it in the climate session.
Have you tried it for North American data where the dating of the arrival of humans is rather clearer than the more drawn-out process in Europe and Asia?
Can you ask me that? I cannot hear you very well.
Sorry, have you tried your analysis for North American data specifically separated from Europe and Asia? Because in Europe and Asia, humans had a very long and extended period of colonization and potential direct hunting, whereas in North America, humans arrived at a more specific time, and therefore you might be able to see different signatures which might allow you to help distinguish whether what's a climate effect and what is a human effect.
Yeah, it's a very good point. I haven't evaluated separately, like only America, only Eurasia. I haven't separated any of the data. It's very hard also because, for example, for the woolly rhino, I only have 55 points, right? And it's only Eurasia. But if I start dividing these in like regions or whatever, I will start having like only 10 points to evaluate population size, for example. And then I will have these huge confidence intervals. So it's a very good point. It's hard to work with that when you have this kind of fossil that is very scattered. And when you start segmenting the data, you will start having holes for response to your hypothesis. It's a extremely good point.
Any more? Your lags in the climate effect look like they're cycling.
Yeah.
Is it seasonality?
No, I think that this is just a fact of how the cross correlation is. So you will start moving the correlation 1, 2, 3, 4 lags, and in some point you will start having the same response because you are moving it in a way that is -1 or +1, and at some point in 5 you will start the same response at 1 because you are doing the full window. Yeah, it looks like Sino.
Well, so yes, and if it was a kind of just a strict moving bid, you'd expect a moving average type of signature.
But what you see,
where you see is kind of much more.
The sinuosity or distance is basically the correlation, not the response. It's how well it fits the correlation.
Time for another if there is another.